To understand the complete details of the cardiac action potential, first, let's take an idea of some important concepts. Where the first one is membrane potential. It can be defined as an electrical potential difference or difference in polarity of charges between the intracellular and extracellular environment across the cell membrane. But how this electrical potential develops across the membrane? Well, it develops because of the difference in the flow of oppositely charged ions across the cell membrane. Let us try to understand this concept through the diagram that is showing the intracellular and extracellular spaces. The normal concentration of potassium intracellularly is 135 millimole, and extracellularly it is approximately 4 millimole, while the normal concentration of sodium intracellularly is 10 millimole per liter, while extracellularly it is 145 millimole per liter. So we see that there is more concentration of sodium outside the cell, while more concentration of potassium inside the cell. And this imbalance occurs due to the leakage of certain ions via ion channels. Let me explain in detail. Here we will talk about cardiomyocytes particularly. There is a sodium-potassium ATPase, or sodium-potassium pump. That is actually an enzyme scattered throughout the cell membrane, and it is responsible for the active transport of sodium and potassium. It utilizes the energy derived from ATP, converting it into ADP and free phosphate. And by utilizing this energy, it transports 3 sodium ions out of the cell, in exchange of 2 potassium ions inside the cell. This phenomenon that takes place in almost all cells of the body in varying degrees results in increased extracellular concentration of sodium ions, the increased intracellular concentration of potassium ions. And as more positive ions are moving outside than inside of the cell, it makes the interior of the cells more negatively charged as compared to the exterior of the cell. That ultimately generates a difference in ionic charge across the cell membrane, generating a transmembrane electrical potential called the membrane potential. This membrane potential varies for different cell types. So until now, we learned that the distribution of positive and negative ions is not the same across the membrane, inside and outside the cell. This difference in ionic charge concentrations, together with the difference in permeability for these ions across the membrane, establishes the resting membrane potential of the cells. Let's define it properly before knowing its further details. The resting membrane potential is the stable electrical potential across the cell membrane caused by ions, predominantly potassium, flowing down the electrochemical gradient that was established by the sodium-potassium pump. Generally, when we talk about ions, we consider sodium, chloride, and calcium. That is higher in concentrations outside the cell. And potassium, which is high in concentration inside the cell. And some other anions that are produced inside the cells, like proteins and amino acids. We have already discussed the sodium-potassium pump. That causes an imbalance in sodium and potassium concentration and develops membrane potential. To understand resting membrane potential, just look at any one ion, for example, potassium, as it is a predominant ion. We see that the concentration of potassium inside the cell is greater than that outside the cell. While to balance its concentration gradient, potassium starts to move out of the cell, as they can't simply be diffused out of the cell membrane, so they move out through some other ion channels called the potassium leak channels present throughout the membrane. Potassium is positively charged, and so inside the cell, it makes bonds with anions. But when it leaks out of these channels, this bond breaks, erasing this sodium-potassium pump to get more space. So the bond between potassium and anions breaks, and potassium leaks out of the cell membrane, leaving behind the anions, as they cannot move out of these leak channels, creating more negative potential inside the cell. This movement of potassium across the membrane is according to its concentration gradient. With time, when more potassium moves out and more negative ions build up inside the cell, this negative potential starts to attract the positively charged potassium ions back again inside the cell. Now this movement is due to the electrostatic gradient. Here the point where the outward movement of potassium due to the concentration gradient, and the inward movement of potassium due to the concentration gradient becomes equal. That point is called the equilibrium potential for potassium, and its value is -92 millivolts. That means at -92 millivolts electric potential, the inward and outward movement of potassium gets balanced. Similar is the case with other ions as well. Each one has its own equilibrium potential. If we talk about only one type of ion, for example, potassium, then the equilibrium potential of that single ion can determine the resting membrane potential of that whole cell. But practically, there are a number of ions. And here, other than potassium, the major ions involved are sodium, chloride, and calcium ions that have their own equilibrium potentials, like 67 millivolts for sodium, -86 millivolts for chloride, and 123 millivolts for calcium. All these equilibrium potentials of ions determine the overall resting potential of the membrane. But other than equilibrium potential, the permeability of the cell membrane for each ion also play a role in determining the resting potential of the membrane. By multiplying these equilibrium potentials of ions with their respective permeability across the membrane, and then adding them all together, one can get the resting membrane potential of that cell. And it comes out nearest to the equilibrium potential of the ion, which has particularly high permeability across the membrane. So by changing permeability, the resting membrane potential also gets changed. So this was a brief concept about resting membrane potential, which is actually a stable electrical potential. Now let's understand an important concept, which is the result of unstable equilibrium potential, and that is the action potential. Starting with its definition, a mechanism by which excitable cells, for example, neurons and myocytes, can rapidly transmit electrical signals along their cell membranes and to the adjacent cells. Here, our focus will be mainly on cardiac action potential. So a cardiac action potential is a change in voltage across the cell membrane of the cardiac myocytes. That in turn excites the heart and ultimately causes the contraction of the heart. Now let's try to understand cardiac action potential via this graph, showing different phases of action potential in cardiac myocytes. Here, we take the membrane potential of a cardiac myocyte on y-axis. The upside of the baseline shows positive membrane potential, expressed in millivolts, and downward showing negative membrane potential, and a time to conduct this action potential on the x-axis. Here the major ions that mostly influence cardiac action potential are calcium, sodium, and potassium. I am writing them according to their respective equilibrium potentials. That is 123 for calcium, 67 for sodium, and I've 92 millivolts for potassium. And the resting membrane potential for cardiac myocytes is approximately -90 millivolts. Here we need to understand. When the membrane potential moves upward and becomes less negative or more positive than that of its resting potential, the membrane is said to be depolarized. Reversibly, when the membrane potential moves downwards or becomes more negative or less positive than its resting potential. It is said to be repolarized. And in case, if it becomes even more negative than that of its resting potential, it is said to be hyperpolarized. Now, to understand this graphical representation of cardiac action potential, let's have a side-by-side look at the cardiac myocyte. Under resting phase, or relaxed state of the heart, the sodium channels that are presented here in green, scattered in the membrane, are usually closed, and they do not allow the movement of sodium inside the cell, despite of its higher concentration outside the cell. This maintains the resting membrane potential of the cells to almost negative 90 millivolts. This state continues for some time. That actually represents the relaxed state of the heart. But as we explained before, to balance its concentration gradient, potassium gets leaked out of the cell through potassium leak channels across the membrane. And thus, it makes the inside of the cell more electronegative. Consequently, to overcome the negative potential inside the cell, some sodium and calcium ions start to move inside the cell through the gap junctions. Although this leakage is very negligible, over time it starts to increase the potential inside the cell, and quickly it reaches up to negative 70 millivolts. We can plot it in the graph, moving in an upward direction. This point is called the threshold potential. At this point, new channels start to open, that are usually sodium channels. These channels were already present there, but were closed before. Now, when the membrane potential reaches its threshold level, these channels get opened. So we can say the threshold potential is the minimum potential for the opening of voltage-gated sodium channels. When these channels open, sodium starts to move inside the cell hurriedly, and the membrane potential inside the cell rapidly moves towards a positive value. Let's say it now reached around positive 30 or so. Here we can see a steep rise in the graph, but then these sodium channels shuts down before reaching the equilibrium potential of sodium at the same speed as they got opened. Now at this point, when the sodium channel is closed, potassium is again predominant now, and like any other ion, it wants to reach its equilibrium state. So more potassium voltage-gated channels get opened. That was there already from the beginning, but was closed. And now when the membrane potential reaches to the positive side, it gets opened. And potassium starts to move out of the cell through these channels. So here, look at the graph again, we see a bit of downward repolarization, and it reaches to positive 10, for example. Now look at the cell again, as this is the point when calcium leaks in. Because now the calcium voltage-gated channel gets opened. Needless to mention, like all other ion channels, they were there from the beginning. They just opened at this point. Here we see that both things are happening simultaneously. Potassium is moving out, and calcium is moving inside the cell. So potassium efflux will try to take the graph downward or towards repolarization, whereas calcium influx would like to take it upward or towards depolarization. So it balances out each other, and we get a straight or flat line in the graph. So for a few milliseconds, membrane potential remains the same. After that, just like sodium channels, calcium channels shut down as quickly as they opened. And now we have just potassium channels opened. So from here, the graph of action potential will start to move downward again, until it reaches the same resting potential. That was negative 90 millivolts. Then at resting potential, the voltage-gated potassium channels will also close down. And now we can see we are again at the same stage from where we began. So after a bit, this slight leakage of potassium starts to happen again, repeating the same exact process. These are actually different stages of the action potential. There are 5 stages of the cardiac action potential. Let us summarize this whole cycle of action potential and name these stages. Here, first comes the stage 4. It is actually the baseline state where muscle cells are relaxed and all ion channels are closed. Only a few potassium ion leaks out of potassium leak channels. Then comes stage 0. That is a state of the rapid action potential when it hits the threshold level. Due to sodium and potassium inward movement through gap junctions, and then reaching the threshold, the sodium channel gets opened. Next comes the stage 1. That is a fast repolarization phase, where the sodium channel gets closed and only potassium channels are opened. Next is stage 2, where calcium channels opening balances the potassium channels. Hence, both calcium and potassium channels are opened. And then lastly comes stage 3, where again, all channels are closed, only voltage-gated potassium channels are opened. Then again, stage 4 comes, and the cycle continues. Also, we can say at the start of the action potential, there is a resting or relaxed phase. Then the inward flow of positive ions causes depolarization. Later, it is repolarized by potassium ions efflux. Then this efflux is balanced by calcium influx, showing a constant plateau. Then the membrane gets repolarized again due to potassium efflux before returning back to its resting state. During this action potential, there is a time duration when no subsequent action potential can occur. Or we can say, during this time period, the excitable cells can no longer produce another action potential. This graph is the most common and typical presentation of the cardiac action potential, but it is actually called the fast response action potential that is passed generally to cardiac myocytes, particularly non-pacemaker cells. There is also slow response action potential as well, that is usually generated in pacemaker cells of the heart, that together form the cardiac conduction system. Here is a graph of slow response action potential. And here we can see that instead of 5 stages, as in fast action potential, there are 3 stages only. As in slow response action potential, there is no resting membrane potential, and the minimum membrane potential of pacemaker cells is usually less negative than negative 90 millivolts, as in non-pacemaker cardiomyocytes. In these cells, there is gradual and spontaneous, but continuous depolarization occurs, which we call stage 4. Then once this gradual depolarization leads to the threshold level, sudden depolarization occurs, leading to stage 0. But it is not that much rapid, as in fast response action potential. Here we see that there is no plateau, and so there is no stage 1 or stage 2, but direct stage 3, where gradual repolarization takes place. When it reaches minimum membrane potential, then phase 4 starts again with continuous gradual depolarization. Here, phase 4 is due to the inward movement of sodium ions. Phase 0 is because of the inward flow of calcium. And last phase 3 is due to the outward flow of potassium. These graphs vary in different pacemaker cells depending on the permeability of a particular ion. Then these pacemaker cells transmit the signal to the nearby non-pacemaker cells through gap junctions, and thus it spread evenly to the atria first, leading to atrial contraction. And then with a slight delay, it reaches the ventricular muscles, as we have explained earlier, causing ventricular contraction, and the process continues. So this was all about the cardiac action potential, where we discussed in detail how action potential is generated and passed throughout the cardiac myocytes. Now moving to the next section.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.